Okay, so in this lecture, we are going to be talking about biological membranes. We're going to start off by doing a little bit of a review of macromolecules and molecules of life in general from chapter 2. And then we are going to specifically focus on two macromolecules this week, fatty acids primarily, and then a little bit proteins, since those two are the main components of biological membranes. And then majority of the remainder of this lecture is going to be focused on the structure and function a little bit of biological membranes. Today is mostly structure, and then next time we will be focusing primarily on the function of these membranes. I'm going to set it up so that I make smaller recordings of this lecture in parts, so you can look at each section kind of a little bit separately. So in this first recording, I will focus on the macromolecules associated with biological membranes and just general chemistry of life. Okay? So when we look at life in general, we find that majority of life is based on carbon compounds. Actually, all life is based on carbon compounds. There are many reasons for that, and we will discuss a little bit about that in a minute. And it also depends. Life also requires water, right? Because majority of the reactions that are required for our survival take place in the aqueous environment, in the aqueous cytoplasm inside our cell. And another requirement of life in general is that majority of life, with some exceptions obviously, the majority of life lives in a very narrow temperature range. A lot of these reactions take place in an extremely concrete, narrow temperature range. It's not we don't survive in extreme cold or extreme hot. Having said that, of course, there are exceptions to the rules, as we all know. Now, when we look at the molecules or our subunits that make us who we are as living things, there will be some commonalities regardless of how big or small an organism is. And that goes all the way from a unicellular organism to a multicellular ones as well. So regardless of what type of cell you're looking at, what type of life you're looking at, you will find commonalities in the type of macromolecules that make us who we are, to make us alive. These molecules are typically polymers, so they're made of smaller subunits that are arranged in chains or in structures to make these molecules to make us. And they are made through extremely tightly regulated processes that we will learn more about throughout the semester as it progresses. So why do we care about, why is life made of carbon compounds? Why is carbon so important? Well, there are many reasons why carbon compounds are ideal for biological life on Earth, specifically. Carbon-containing compounds are extremely stable. The main reason for that is that a single carbon can make 4 individual bonds, up to 4 individual bonds. That allows it not just variability in the type of compounds it can make, but also stability in the structures that it develops through these bondings. So because of those structures that it develops that are highly stable, the visible light does not have enough energy to break these bonds, and instead you would need either UV light or something stronger, a stronger force to break them. That allows more stable structures to form and allows for life to happen, which is a slow process in some cases. It also, like I said, provides options for a lot of options for the type of molecules that can be created, because there are more binding sites, essentially more interactions that can happen, and those interactions can then allow for greater variability compared to some of the other options out there. When we look at the type of compounds that we are going to see in life, you can actually divide them into two kind of functional groups based on how soluble they are and how reactive they are, how they react with other agents around them, other molecules, other environmental agents around them. So what typically you will see is that majority of molecules that make life, the carbon compounds that make life, are carbon-carbon compounds with hydrogen as their other secondary component. So they are hydrocarbon skeletons essentially. And then they would either have oxygen as an additive to these structures to make them the next level, or they will have nitrogen added to their structures to make them their next level. So when we look at sugars, for example, they will have the oxygen as the third component in them to make many of those compounds. All the alcohols, the aldehydes, ketones, many of these compounds you will see in metabolic reactions as either intermediaries or as reactants or products are going to be carbon-oxygen compounds. On the other hand, you have amines and you have nucleotides that make up your DNAs and DNA, RNA, amines that make up your proteins. Those are nitrogen compounds. So they will contain carbon and nitrogen along with the other agents that you have. Okay. So these are the two categories that you should be aware of: your carbon-oxygen compounds and then the carbon-nitrogen compounds. Now, along with these compounds that you're developing, you would also need an environment for these compounds to be housed in, to be hanging out in. And for that, what better thing to use than water? Since water is considered, as we've known before, even in chemistry, a universal solvent. It becomes equally important in biological systems because water can form spheres of hydration around ions to kind of keep them in place. It can also interact with a variety of agents, both small and large compounds, both small and large, and help them become dismissible and movable within the cell environment. So majority of life actually depends on water as its main solvent within their system. Now, there are two categories of compounds that you're going to see within biological systems. Just like any other place, you'll have hydrophilic compounds that are water-loving and can easily get dissolved or mixed and interact with water molecules. But then you're also going to be looking at hydrophobic compounds that are water-fearing and that would not interact readily with water environments. Kind of like what we're going to be looking at today, the lipids that make up our biological membranes. These hydrophobic compounds have a very important role in maintaining our cell's environment. That's why we can create those compartments that allow for the cells to do the work that they need to do, that allow the cells to isolate these lysosomal environments or peroxisome, or these environments that are going to be extremely harsh for the cell as a whole to survive in, and do the work that it needs to do in that specialized setting. So hydrophilic compounds are hydrophilic because they either are ionic, so these are, like sodium ion, chloride ion, they have a strong positive or negative charge, or because they are polar. And that's because the way they have hydrogen bonds within them that are able, they have hydrogen and oxygen within their structure that is able to form hydrogen bonds with oxygen or the hydrogen from the water molecules. So because water is a polar molecule where it has a partial positive or partial negative charge, it can interact with other polar molecules quite easily because of their partial positive or negative charges. So for an object to be hydrophilic, for a molecule or a substance to be hydrophilic, it should either have a charge, a positive or a negative charge as a whole, or should be polar, where it has partial positive and negative ends. Hydrophobic compounds, on the other hand, will generally be non-polar, so they are going to be insoluble in water, right? They won't be able to be dismissed inside, mixed up in the water as a whole. And that is partly because of their structure, the way they are built, right? And typically they will have a lot of carbon hydrogen, so there'll be a lot of hydrocarbon chain structures, and those will be typically hydrophobic when they are just carbon and hydrogen with no other substance, no other atom involved that can provide it with that polarity needed to interact with water molecules. In that case, usually what you will find is that those nonpolar molecules will tend to stick together, while the water would also tend to stick around it in a way that it minimizes their interaction with each other. So when we look at chemistry of life and look at the small and large molecules that are present inside our living systems, there are certain small molecules that we want to focus on. And specifically, there are 4 major categories of compounds, small organic compounds, that we are going to be focusing on. So the first one of those would be sugars, which are both energy providing source as well as energy storage units. And they are going to be your subunits for polysaccharides, long chains of sugar molecules that can act as structural components as well as storage components. A second thing that we want to focus on, like we will today, would be fatty acid chains, which are the main component of biological membranes. And then the third ones that we are going to be looking at are amino acids, which are going to be smaller subunits of proteins that are your worker bees that are doing all the work that you are doing inside your cell at any given time. And then finally, we are going to be looking at nucleotides, which are your subunits primarily for DNA and RNA. Now, sugars, fatty acids, as well as amino acids, all 3 of them can act, as storage molecules. Nucleotides are typically considered as, storage molecules for energy. That is. Nucleotides, on the other hand, are mostly thought of as a storage molecule for information in the form of DNA and RNA. However, they do have a very strong energy storage component as well, as in the form of ATP and GTP, which is the basic currency of our energy producing currency inside our body. That's what we use to utilize energy that has been gotten from the sugar, fats, and proteins that we utilize in food. So the 4 major families of small organic molecules that we want to be familiar with and that we want to know about are sugars, fatty acids, amino acids, and nucleotides. We should know what these individual building blocks make inside our body. So the sugars would be creating large organic molecules in the form of polysaccharides, glycogen, and starch. Glycogen would be storage unit of energy inside primarily animal cells, but also some plant cells. Starch is primarily for plant cells. Fatty acids, which will provide us with fats and membrane lipids that create a lot of our membrane structures, obviously, but then also provide storage unit inside our body for energy. Amino acids that are your worker bees that make your proteins that do all the enzymatic activities and work inside our body, make us who we are, look like what we are. And then the nucleotides, which are your nucleic acids in the form of DNA and RNA. And then obviously the energy storage, and there would be the ATP and GTP. When we look at the chemical composition of a cell, we actually find that majority of the cell's weight comes from water molecules. So anywhere from 60 to 80% of any given living cell is going to be just plain water, as it is required for almost all our chemical reactions happening inside the body and to act as a solvent for the various components inside us. The remaining majority is going to be made of these macromolecules that we just talked about. The nucleic acids, proteins, polysaccharides, right? And then the minor components left behind are what are the precursors that make up these macromolecules at the end of the day. So today we are focusing on fatty acids, since that is going to be the main component of biological membranes. So to begin with, let's talk about what is a fatty acid itself. A fatty acid is a carbon-hydrogen chain with some kind of hydrophilic carboxylic acid head. So here you see that carboxyl group on one side, that is the hydrophilic head, because of the presence of oxygen. That's going to give it a partial negative charge on one of the oxygen. And then it has that hydrophobic hydrocarbon tail that's going to not want to interact with water molecule. This is a single chain of a fatty acid. Because it possesses both hydrophilic and hydrophobic region, it is what we call an amphipathic molecule. When we look inside our cells, we see fatty acid chains in various forms doing various types of functions. One of those is storage of lipids inside our cells and inside our body in general as energy reserve. These are done through the presence of triacylglycerols. Triacylglycerols are built of 3 fatty acid chains that are linked together with a glycerol molecule. Hence, triacylglycerols. Tri meaning 3 fatty acid chains and attached with the glycerol molecule. Here is the glycerol molecule. You can look at that. Again, it is made of carbon, hydrogen, and then some oxygen molecules that are used to link the fatty acid chains together through an ester linkage. These are used to store lipids inside our cells. Now, these can be found in various forms, however. Not all triacylglycerols are going to look exactly the same. They are going to vary depending upon the type of fat that is used to create the fatty acid chains, which can be either saturated or unsaturated. And they will also be different depending upon the length of the fatty acid chain, whether it is long or short. Typically, there is a small range of fatty acid carbon-hydrogen chain, carbon chain that is built. It's usually between 16 to 21 carbon long, but it can be longer or shorter depending on the needs of the cell or the type of the fatty acid glycerols that are being built. So here is an example of a saturated fatty acid tail. In this case, it is a fully saturated carbon-hydrogen chain, so there are no double carbon-carbon bonds. It's a single carbon-carbon bond with all the other bondings taken over by the hydrogen atoms. On the other end, you have an unsaturated fatty acid tails over here, example of that. And you notice that they have these little kinks. They have these bends in the tails. Anytime there's a bend, that indicates that there is a double carbon-carbon bond, which is slightly rigid, that kind of holds that area in place. And then the next carbon bond is going to be a little bit to the side one way or another. This gives this type of triacylglycerol a little bit more bulky appearance because of the kinks. It's not going to be packed as tight because it doesn't hold straight lines, right? So it's kind of like trying to stack pencils straight. If you have straws or pencils that are completely straight, unbendable, they're rigid, you can stack more of them together. You can pack them tighter in a container versus if they have bendy ends, or then you won't be able to pack as many because they're going to have those little kinks involved. That's the same idea here. Now, in our membranes, you have very similar looking fatty acid chains, but these are going to be different from the triacylglycerols in the sense that they will have a phosphate group attached at the end. This phosphate group is what gives them a highly polar end, right? It is going to have this highly hydrophilic head, and there are only going to be 2 fatty acid tails instead of 3. Again, they can come in various sizes: small, short, short, long chains, saturated or unsaturated, but they will always have 2 fatty acid tails attached with the same glycerol that is then attached to a phosphate group. So they are phospholipids that make up our biological membranes. Now there are additional spaces. There's an additional group that can be attached to the top end of this phosphate, and that will change depending upon the needs of the cell, the type of phospholipids that you're trying to create. One of the things that can attach to that third point of attachment is going to be a small choline molecule, right? So that is one possibility in the type of attachments that can be made. There are others that can be made as well, and I'm going to show you some of those examples in a few minutes as well. So here is an example of how then one of these phospholipids will interact with others to create a lipid membrane, right? So here is your typical phospholipid molecule. It contains two hydrophobic fatty acid tails. It has a glycerol that is attaching those two together. It is then followed by a phosphate group that has an attachment point to attach a third molecule on top. That is usually a polar group of some sort that is going to keep that entire system in a way that there's a highly hydrophilic head and a highly hydrophobic tail. Now, if you were to mix some of these in water, right, it is going to be a highly energy unfavorable condition. So to get into a more favorable condition and try to exclude water as much as possible from their system, they would automatically tend to line up in a way that the hydrophobic tails interact with each other and the hydrophilic heads are what face the water molecule on either side. So they end up creating a bilayer spontaneously in these conditions. That is how you get your membrane, essentially. Now, what type of modifications, we talked about that polar head, can be made on that phosphate group? Well, there are several different types that you can do, but there are two main categories, okay? So one of them is what we call phospholipids. These are where you have some kind of a group attached to that phosphate that enhances its function. So for example, a choline group, a serine group, an inositol group, all those would be part of what is attaching directly to that phosphate to enhance its function, or what we call sphingomyelins. All of these are extremely important in specific parts of our body in their biological function they perform. And we'll be talking about those more in detail as well. On the other hand, there is another type of modification that can be made to phospholipids that's very different. It doesn't enhance its polar nature, but it is extremely important in cell signaling and cell-cell interactions. And that's when there is a sugar molecule attached to your actual fatty acid chains, the main phospholipid. In that case, it can be attached directly to the glycerol, or it can be attached to a phosphate group as well sometimes. But many times it's a direct interaction with that glycerol on the fatty acid chain. So in this case, these are called glycolipids. So phospholipids, you have the phosphates that are then interacting with other molecules, right? And glycolipids is where it is the glucose molecule, not glucose molecules, a sugar molecule. It doesn't have to be glucose. It can be any sugar related to the function. It's a sugar molecule, O-glycan, N-glycan, doesn't matter. It depends on the type of cell you're looking at, the environment you're looking at. But it is interacting directly with the glycerol. Okay, so that's a very important difference that we want to think about in this state. Now, membranes are going to act as selective barriers, whether they are outer plasma membrane or internal, compartmental membranes. So in bacterial cell, you're only going to have that outer plasma membrane. In the eukaryotic cell, you're going to have the plasma membrane, and then you're going to have that internal membrane. Inside, for the various organelles, each one of them is going to have its own selective barrier. So obviously, if the barriers are different, that means the plasma membrane is somehow different in those environments as well. So what is it that maintains that selective barrier? Depends on the type of those molecules that are attaching to your phospholipids. And we are going to talk about some of those a little bit later in this lecture. Okay. Now cells are going to interact with their environment outside through the plasma membrane, right? So they're going to use that plasma membrane to receive information, whether it is through a signaling molecule or just interaction with the general environment. They're going to use that plasma membrane to control what goes in and what goes out of the cell, right? So for the import and export of the small molecules. And they're also going to use it in order to move in their environment. So it allows them the ability to interact and move through their environment, either by expanding or actual physical movement. The internal membranes, on the other hand, have very different functions. They are not interacting with the outside environment directly. They are maintaining their own setting, right? Their main goal is to maintain their own selective barriers. And then in addition to that, they are going to be responsible for interacting with the immediate cytosolic environment, right? Their internal environment of the cell. And they're going to be interacting with that and communicating with that in a somewhat similar fashion to what's outside. The internal membranes in a eukaryotic cell, there are many of them, obviously. There's the ER, your nuclear membrane, you have the lysosomes, peroxisomes, the endosomes. There are a lot of different types of membranes floating around there, each one with its own very concrete function. Now, if you think about the endoplasmic reticulum and this Golgi apparatus and the transport vesicles, they may share a lot of the same membrane biology, right? They may be very similar because they are part of that transport system. But a peroxisome and lysosome is specifically, right, which have very different environment inside, internally, that needs to be maintained for their appropriate function, are going to have a very different cell biology of that membrane. Their membranes are going to look very different than what you see on those other membranes. Same thing with the mitochondria. Because those are much hostile environments internally, they're going to have a specific biology to maintain it, to stop that from getting outside, to maintain the integrity of the cell as a whole. Now, part of the way that especially the biological membranes, the outside membranes, but also the nuclear membrane and other membranes interact with each other and interact with their environment and communicate signals is through presence of these of proteins within them that are able to provide channels and transport systems for signals to pass through to interact with outside environment and inside cytoplasm. So we are going to be looking at each one of those components in the next part of our lecture. Okay, so I'm going to stop this one at this point, and in the next part of this lecture, I will go further in detail about how the various lipid membranes, various biological membranes maintain their biology, how they are different when they are outside in the biological system, in the plasma as a plasma membrane, versus how they are internally when they're facing cytoplasm or when they're part of another organelle that has a very different environment. And then in the third part, I will focus on what type of proteins are going to be present inside the lipid membrane and how each one of them is going to be different in its structure and in the way that it interacts with the plasma membrane and the environment. Okay?